All right. Good morning. It is Wednesday, April 21st, 2020. We are on to our last lecture of part 4. We get to finish up the, apparently I misspelled urinary system there, the urinary system. And we are going to finish our talk of the physiology of that. And then I'm hoping there will be time for a review at the end of it. Shouldn't take the whole class time. So hopefully we'll have a chance for a little Q&A review. If you guys have any questions, and then that's all leading up to Monday's exam, lab and lecture. Then you do have a pre-lab activity, the Labster, and then we come back, 4 lectures on the reproductive system, and then final lab and lecture exam, and then the final, and then we're done. So we are rapidly approaching the end of all of this. All right, questions on any of that? All right, let's dive in then. All right, we were at the end of last class. We had finished pretty much everything that we needed to know about the renal corpuscle and the glomerular filtration that takes place there. As we know, that is where we produce those 200 liters of filtrate during the course of the day, and then it has to be processed. And from that Bowman's capsule, remember, it goes straight into the proximal convoluted tubule, which, as we said, is where the majority of all of the processing takes place. So there's a tremendous amount of new information here, but I do want to talk a little bit about what is going on here in the proximal convoluted tubule. The first thing that happens is we get the reabsorption of organic nutrients, right? This is things like glucose, things like amino acids, things like nucleic acids, all of those types of things. And essentially, in our proximal convoluted tubule, 100% of this stuff is reabsorbed. Here in the proximal convoluted tubule. So all those things like glucose, like amino acids, like all those types of components that we need, they are going to be reabsorbed. This is, again, primarily done through active transport and co-transporters, but there can be other types of facilitated diffusion. Facilitated movement like pumps, and also like even some passive channels and carriers and things along those lines. But it's more than just the organic nutrients. We also have to actively, again, using ATP, absorb a vast majority of the ions as well. We are going to be actively pumping sodium back. In fact, that movement of sodium, as we're going to see, is going to be vital for helping to facilitate the movement of a lot of the other materials like those organic nutrients as well. Chloride, phosphates, magnesium, all of those types of things are actively transported back. However, remember, as we talked about, it's not going to be as important for potassium and also calcium. While obviously it is important to have calcium and potassium in our blood, in our interstitial fluid, for those, it's really about having the appropriate amounts, and the appropriate amounts of those have to be carefully regulated. Potassium levels we want relatively low, so that we can maintain that potassium concentration gradient from outside the cell to inside the cell. And calcium, remember, we have that very narrow range because calcium makes cells do wonky things. Now, notice when we're moving all of this stuff from the filtrate back into the body, right? That's a lot of stuff that's moving. And what likes to follow stuff?
Water.
Water, absolutely. Oops. There we go. Water. Absolutely. So as we're moving all of these things back, remember we talked about water follows salts, or water follows stuff. And remember how we talked about these cells of the nephron can have aquaporins, those water channels that are expressed on their surface. Now, when all this stuff is moving back, water has to follow it, right? Because water has to go where stuff is. We call this obligatory water reabsorption, right? Obligatory means that you have to do it, right? My brother-in-law has a birthday and I have to go to his party, right? Again, I'm required to do it. I don't have a choice in the matter. I'm obliged to go. And that's the same thing with water as well. Here in the proximal convoluted tubule, water has to follow all of this stuff. All right, so again, if you think about this, let's go ahead and draw this because I want to make one more point on this. So here we'll put two of the cells, of the absorptive cells here in our proximal convoluted tubule, which, as we talked about, have those tight junctions between it. And so again, we've got all this movement of sodium, of glucose, of other ions, and all these things that are going, all this stuff that is moving, that is getting drawn back into the body, and therefore water is forced to follow as well. Oops, don't hold on, that can stay black. I want my arrow to be blue. Water can follow as well. So we have this obligatory water reabsorption. Now, this obligatory water reabsorption is obviously important because we need to retain most of that water that is going to come as well. But notice also, as this water travels back into it, if we're over here inside the lumen of our tubule, as water is resorbed, we still have a stuff that remains behind, but water leaves. As that water leaves, that is going to concentrate the remaining materials. And that concentration, oops, if I spell concentrate correctly, sets up a concentration gradient. And that concentration gradient can help to drive the passive paracellular reabsorption of materials. So as the stuff here in the lumen gets more and more concentrated, we get a stronger and stronger driving force for this. And as a result of that, many components are actually able to follow, oops, that should be green. Follow that concentration gradient from the denser concentration here inside the lumen between the cells back into the body. And again, if it's going paracellularly, then obviously there are no transporters involved, and this is a passive process. So we're using just that concentration that is being set up by that obligatory water reabsorption to move things back into the body. And so, of course, that is our fourth major process, that passive reabsorption of ions, moving down their concentration gradients, and many things, again, paracellular, or in some cases, leak channels as well. And for instance, for some micronutrients, for instance, and some ions, as much as 50% of those are actually reabsorbed passively using this process. Now, notice all of this stuff involves substances and water moving back into the body. But remember also the proximal convoluted tubule is where also most of the tubular secretion takes place as well. Again, one of the points that I wanted to emphasize is these are both happening at the same time. It is not sequential. We don't do all the reabsorption and then all of the secretion. These are both moving at the same time. We are just talking about them separately because it makes more sense to do it that way. But these are all happening at the same time, primarily in the proximal convoluted tubule. All right, let's talk about some of the pathways involved in this. As I mentioned, one of the most important ones is sodium. As we learned way back in 430, sodium is the most common cation in the body. There's a large concentration of it outside of cells. There is it's a positive ion. The inside of the cell is negative. Rest of membrane potential is negative 70 millivolts. So there's a huge driving force of sodium. And as we've talked about, we can use sodium to do work. So our sodium transport is important for two things. Obviously, we need to get that sodium back into the interstitial fluid, into the blood. But at the same time, we're also going to be able to use the driving force of sodium to do work for us, primarily in the form of co-transporters. So understanding this pathway associated with sodium, the movement of sodium, is going to be really, really important. All right. Actually, you know what? Let's do it this way. Let's go ahead and draw this. I'm just going to draw the cell as a square. No, it's not a square. There we go. So here are our cells. Again, just for clarification, this is the lumen. Oh, that's huge. This is the interstitial fluid. This would be the apical surface. This is the basal surface. So when we talk about sodium, and I think sodium is red, so let's go sodium, red. When we talk about sodium, as we mentioned, it is going to be very, very important to be able to get this back. So on the apical surface, whoa, let's not do that. On the apical surface, we are going to actively transport sodium across the apical surface. This is going to come in the form of both pumps, which of course are primary active transporters, but also co-transporters as well. For right now, let's just draw one of these pumps. So we'll get a little circle. As we know, pumps, of course, use ATP. To move some substances across the apical surface, bringing it in. Again, this isn't that dissimilar to what we saw in digestion as well. We want to get things out of the lumen, because remember, anything that stays in the lumen is lost in the form of urine, right? The same way that anything that stayed in the lumen of our digestive system is lost in the form of feces. So obviously, we want to do all that transporting on the apical surface. Then when we got to the basal surface, we didn't really care about how it got out of the cell on the basal side. We could just let that happen passively and not waste any energy on it. But that's not going to be the case here with sodium. With sodium on the basal surface, we are also going to use ATP, active transport, on the basal surface. Primarily using your friend and mine, the sodium, potassium, ATPase, the sodium potassium pump. Someone remind me again what the sodium potassium ATPase does.
3 sodium in, 2 potassium out.
So 3 sodium, but if we're going to use ATP to move it, do we really need to use sodium to get it to get sodium into the cell? Which way are we going to move those 3 sodium? Yep, absolutely the right idea. We want to move 3 sodium, but which way do we want to move those 3 sodium? Into the cell or out of the cell? Well, sodium wants to go in the cell. So we typically don't have to use ATP to do that. But in this case, we are going to use ATP to kick it out. So we let that kick that sodium out of the cell, because ATPases kick 3 sodium out and bring 2 potassium in. However, this channel has, this basal surface has some potassium leak channels. So basically, as soon as that potassium comes in, that potassium is able to sneak right back out again. Now, the advantage of this setup, the fact that we're using ATP both on the apical and the basal surface, is as soon as sodium comes into the cell, we can quickly rush it back out again. And the advantage of that is we are going to produce an extremely low concentration of sodium inside of these proximal convoluted tubule cells. And as we know, sodium is one of those things we can use to do work for us. And so this is going to set up a huge concentration gradient with the lumen. And that driving force is going to allow us to help to bring other materials in. So not only is this important because we then get sodium back into the interstitial fluid, and of course, once it's in the interstitial fluid, it then travels into the blood. Let's say it this way, the peritubular capillaries. But it is going to set up a huge concentration gradient that is going to allow us to do other work. That work, for instance, is going to help us to reabsorb our organic nutrients. So all of those organic nutrients that we talked about are going to be important for reabsorption. And they are primarily going to be brought across the apical surface using one of those secondary active transporters, using a co-transporter. Using a co-transporter with sodium. So again, now that we set up this concentration gradient, sodium really, really, really wants to come into the cell. And so we say, okay, sodium, you can come into the cell. But when you do that, you have to do some work for us. You're going to need to bring something else in with it. That something else is going to be something like a glucose or an amino acid or nucleic acid or any of those other types of organic nutrients that need to be brought back in. Now, of course, once that sodium comes in, it needs to be kicked out again. But as we know, we have these pumps over here that is going to do that work to get rid of that sodium. And those glucoses, those amino acids, again, now once they're in the cell, they're not going to be lost. So in those instances, primarily they are going to leave via just facilitated diffusion. All right. Questions on those? One more quick point. Remember this co-transporter that moves things in the same direction. Remember, we call a symporter. I make a point of emphasizing that because we have one more important pathway that we need to talk about, absorption, reabsorption pathway. And this involves a dual combo thing. This is about the reabsorption of our friend bicarbonate, but also at the same time, this involves the secretion of hydrogen ions. Why is the reabsorption of bicarbonate and the secretion of hydrogen ions so important?
Regulating the pH level of the body.
Exactly. This plays a huge role in regulating our blood's pH. So let's talk about this process, this pathway. It starts this time with an antiporter. This antiporter oops. So again, it's a co-transporter with sodium, but this time it's an antiporter. And so what happens is we, again, let sodium into the cell, which of course, as we know, then needs to be kicked back out. And when sodium comes in, it actually kicks out a hydrogen ion. Now, again, this has accomplished our goal of getting the hydrogen ion out here into the lumen so that it can be left. But remember, we have this bicarbonate in here as well. And if I remember correctly from when we were doing the respiratory system, there is an interesting relationship between hydrogen ions and bicarbonate ions. And what is that? When you get too many hydrogen ions and bicarbonate ions together, what happens?
Breaks down into water and CO2.
Yeah, absolutely. You're right. Absolutely. There is an intermediate step. Remember, the two come together to form carbonic acid. But you are absolutely correct. That carbonic acid can then dissociate into water and carbon dioxide. Right? Remember, we talked about how this is a reversible chemical reaction. But notice out here in the lumen, oh, that was sweet. Here, there is an increase in hydrogen ions. And as there is an increase in hydrogen ions, that is going to drive the chemical reaction in this direction. So as hydrogen is pumped out of the cell into the lumen, that increases the hydrogen concentration and it drives this chemical reaction to the right. And so as a result of that, we get water and carbon dioxide. Now, carbon dioxide is a small non-polar gas. And as it starts to build up in the lumen, it produces a pressure gradient. And as we know, that partial pressure gradient, it goes down its partial pressure gradient. So it's going to want to leave the lumen. And that carbon dioxide is going to enter into the cell. And what does carbon dioxide need to get inside of the cell? Does it need a pump? Does it need a carrier? Does it need a channel? Does it need a transporter? What does it need?
Probably need a pump, right?
Well, remember, it is small and it is nonpolar. Did it need a pump in the respiratory system or a transporter? No, it just got through by simple diffusion. And guess what? That's exactly what's going to happen here. Carbon dioxide enters the cell via just simple diffusion. All right. Oh, I don't know if I made the cell big enough. I'm going to have to cheat here. So now we are getting an increase. Here, let me put this down here. In carbon dioxide inside the cell. Why might that be significant?
That CO2 doesn't belong there.
True, CO2 doesn't belong there, but what else is inside this cell?
Oxygen.
Oxygen's inside the cell, true, absolutely. What else is inside the cell?
Water.
Water. And what happens when you mix CO2 and water together?
The reaction goes the opposite direction.
Exactly. I'm going to cheat and make my cell a little bigger here. That's still not the right size. Absolutely. We are going to have that same reaction, right? However, this time it is the CO2 levels that are increasing. And as that CO2 level increases, that is going to drive our chemical reaction in the other direction. So that CO2 is going to mix with the water that is inside of the cell. And as that occurs, it is going to form carbonic acid, which will then dissociate into hydrogen ions and bicarbonate. So this time it is an increase, this time it is an increase in carbon dioxide that drives the chemical reaction in this direction, producing hydrogen ions and bicarbonate. Once we get hydrogen ions, we can then kick them back out of the cell again, and the process can continue. Of course, we can't let the bicarbonate build up inside this cell either, because it would start driving the chemical reaction in the other direction. So we got to take that bicarbonate and we need to kick it out into the interstitial fluid. And notice as a result of that, basically what is happening here is we are able to reabsorb our bicarbonate, get that important pH buffer back into the interstitial fluid and into the paratubular capillaries, and we're able to kick out hydrogen ions, being able to maintain our pH in our blood. I think I've done an okay job of showing you this here, but let's look at these with the pretty pictures from your textbook. Notice if we look at the pretty pictures from your textbook, again, as we talked about, we are actively transporting sodium into the lumen of the cell. Into the cell. We want to get it out of the lumen because we need to save and retain that. But we're also going to actively pump it out to the basal surface as well, using that sodium-potassium ATPase, because we want that very, very low concentration of sodium to be able to drive much of the other transport that we want to take place. Of course, as we mentioned, this ATPase also brings a lot of potassium in. And so that could end up upsetting the balance of potassium inside and outside the cell. But remember, we have those leak channels that let the potassium go right back out again. So our potassium levels stay normal, even though our sodium levels get very, very low. So again, keeps that very low intercellular concentration, which produces a strong gradient we can use for our co-transporters. All right, questions on this sodium transport? One of these three transport mechanisms is very likely to be an essay question. So again, let's make sure we understand this as we move forward. All right, so as I mentioned, we can then use this to do work for us, like the reabsorption of our organic nutrients like glucose, amino acids, and things along those lines. Notice, as we talked about, they come in via a co-transporter of sodium. So sodium comes in, that glucose or that amino acid comes in with it. And remember, they're moving the same direction. So this is a symporter. Again, that sodium, when it comes in, needs to be kicked right back out again, so that sodium can continue to do work. But once we've saved the glucose from the lumen, we don't have to be quite as concerned about it. And so we can just rely on facilitated diffusion to get it out of the basal surface. Remember, we also at the same time talked about we need to do that, that hydrogen secretion, but also that reabsorption of bicarbonate. Although technically it's carbon dioxide that is actually reabsorbed, but the advantage of that is that we get the bicarbonate. Notice again, as we talked about, we use an antiporter where sodium comes in and it kicks the hydrogen out. When that occurs, we get an increase in hydrogen ions inside the lumen of our tubule, which drives this chemical reaction to the right, producing a large amount of carbon dioxide, which is able to just pass through the plasma membrane by simple diffusion. But essentially what that does is increases the amount of carbon dioxide in the cell, where it interacts with our water, makes our carbonic acid, which dissociates into hydrogen and bicarbonate. That hydrogen can then be pumped right back out, continuing the process going. And again, because we want to maintain the concentrations, we are going to actively pump this out. And one of the points I want to make with this illustration right here, this is not a co-transporter. This is just a simple pump. It is using ATP to move both sodium and bicarbonate. Remember, sodium isn't going to want to move out of the cell by itself. So this is a pump using ATP to directly move sodium out of the cell and move bicarbonate out of the cell as well. All right, excellent. So like I said, one of those three pathways are likely to be an essay question on the exam. And here's one more pretty picture showing all of these pathways that we were just talking about. All right, summing up our reabsorption in the proximal convoluted tubule. Questions on that? All right. Again, your book's got some nice tables that does a good job of talking about some of these mechanisms involved in the reabsorption of things as well, other than those three we talked about, but those three are the ones that are going to be the essay questions. All right. So not surprisingly, by the time our filtrate has reached the end of the proximal convoluted tubule, the condition of it has changed dramatically, right? 100% of the glucose, 100% of the amino acids, right? 65% of the sodium, 65% of the water, right? 60% of the chloride, 55% of the potassium, right? And remember, potassium reabsorption isn't as important. And so on and so forth has taken place. The volume has changed dramatically. We're at practically a third of what we started with. And a lot of the other materials are gone as well. Basically, what we're left with at this point is water, wastes, and ions. Whoops. Right, these are the things that are pretty much left at the end of this process. But there's still a lot of water. There's still a lot of ions that need to be reabsorbed. And that is going to be one of the major functions of our loop of Henle, where we're going to go next. All right. However, I do want to briefly mention our tubular secretion. It's pretty simple and straightforward because, as we talked about, it's using the exact same type of mechanisms, active transporters, code transporters, facilitated diffusion, things along those lines. The main difference is it's basically just going in the opposite direction, right? In the case of tubular secretion, we are going from the blood to the filtrate. Whereas in reabsorption, we're going from the filtrate to the blood. So the mechanisms, the types of transporters, the types of pumps are the same. What's different is the direction, and obviously the other big difference is the materials that are moved, right? In this, we're removing substances that we don't want, toxins, excess amounts of ions, hormones, all sorts of materials like that that are produced by the body are being released. Neurotransmitters can be released from the body this way. The other thing too is, remember, as we talked about in the proximal convoluted tubule, there is that concentration gradient which is set up, which allows for that passive transport via paracellular pathways. And again, when it's passive, it can't be selective. What that means is things, small, in particular, small wastes with a high concentration gradient are able to sneak back in. Two in particular, urea and uric acid, are very small nitrogen-based wastes that often come back in via that paracellular reabsorption, and as a result need to then be actively pumped back out again. Urea in particular is a very, very interesting biological molecule. It is a metabolic waste. But also, if you remember, carbon dioxide is a metabolic waste as well. And is the goal of our respiratory system to get rid of all of the carbon dioxide in our body? Because carbon dioxide is a waste, so it must be bad. No, exactly. Because it plays such an important role in maintaining pH. We need to have low levels of it. We need to control those low levels of it, but we don't want to get rid of all of it because it plays an important role in regulating the pH of our blood. Well, urea is the same way. Urea is indeed a waste, but we don't want to necessarily get rid of all of it because urea plays a huge role in helping to maintain the osmolarity of the blood. Remember, osmolarity is the measure of the amount of stuff that is in the blood. And so urea, again, we want to have the appropriate levels. We want to have low levels, but we don't want to get rid of all of it. So it has a very dynamic cycle of being able to come back in and be kicked out and come back in and be kicked out and so on and so forth. It's very, very interesting. In fact, the other interesting thing about urea from a biological standpoint is it was the first biological molecule that was produced in a laboratory. I know that doesn't necessarily sound like a big deal, but for many, many years, you know, in not even ancient times, even as late as the early 1900s, it was believed that there was a particular essence in living material that made it different from non-living material. Right, there was some kind of essence, not quite a soul, but some type of living essence that was found in biological materials that you didn't find in non-organic, non-living materials. And so when we were able to, in a laboratory, produce a biological component like urea, that completely shattered that idea that there's some inherent characteristic or feature within, sense of life within living things. So again, it's a very interesting story, but again, we're not gonna have the time to go into it, and you don't care, so it doesn't matter. But the point is that all of this movement is going on in just the opposite direction. All right, so again, removing excess potassium, controlling the pH. We've seen that and talked about that when we were talking about hydrogen ions and the bicarbonate and all that. What I want to talk about now is our loop of Henle. This one takes a little bit of work. So I'll tell you what, even though it's a teeny bit early, let's go ahead and take our first break here so we can come straight fresh to the loop of Henle and make sure that we understand the concepts involved in this. So let's go ahead and do that. It is 8:52 right now, so let's come back at 9:07. And at 9:07, we will restart and we will discuss the counter current mechanism that takes place in the loop of Henle. I'll start the recording at the time. Any questions before we take our first break? All right, see you guys in 15 minutes. All righty. We have moved our way on into the loop of Henle. Again, as we mentioned, close to 2/3 of our filtrate have been reabsorbed, but we still have a lot of sodium and other ions, water and ions that need to be reabsorbed as well. Now, one of the issues, as we've talked about, that we've had is that we've had that obligatory water reabsorption. Basically, everywhere that the solutes go, water is going to follow. And while that's fine as a good driving force for lots of things, we don't necessarily want our urine to have the same concentration of ions in it that our blood and our filtrate does. So we need to be able to decouple the movement of water and solutes. And that is something that we're able to do here in the loop of Henle. This loop of Henle is also important because basically it works in a counter current mechanism. Anybody know what I mean by the term counter current mechanism?
Opposite instead of concurrent?
True, absolutely. By counter current, we have basically what we have is two materials or substances or something along those lines that move in opposite directions of each other. Probably the classic example that many of you might be familiar with is a heat sink. Right? With a heat sink, basically what happens is you have like some kind of this is something that they use in like nuclear power plants, for instance, where we have some really hot like water or hot material that is traveling in one pipe in one direction, and over the top of it in the opposite direction, they move cold water. It's a great question. I don't know. Yeah, they did used to, but as they've gotten smaller and smaller, I think they may have moved away from that. But again, not my area of expertise at all. But you've absolutely got the right idea. Basically, by having things, whether it's air or whether it's water moving in opposite direction, what ends up happening is this cold water basically takes the heat from the hot water as it travels through. And so as a result of this, when the water leaves the other end of that tube, it is now cool water. And again, that heat has been taken from it as it goes across it. So that's that countercurrent mechanism. So we're going to have substances moving in opposite directions, filtrate moving in opposite direction, right? First down the descending loop and then up the ascending loop. And there are two important rules that we need to know and understand about this process. On the descending limb, it is permeable to water, but not permeable to solutes. Whereas in the ascending limb, it is permeable to ions, but it is impermeable to water. Let's go ahead and draw this and then write this out. All right, we can clear this. We're in our loop of Henle. And remember, as we've talked about, let's do this. I'll move this there. Excellent. Yes, thank you. This is our ascending limb. Remember, it is made of a simple cuboidal. Cells, and as we mentioned, it is going to be permeable to solutes, impermeable to water. There, put that there, and just for the heck of it, we will draw some cuboidal cells along the wall of this here. Boom, boom, boom. Excellent. Whereas over here we have the descending limb. That is simple squamous cells. It is permeable to water and impermeable to solutes. Oh, and let's cheat and do one more thing. Connect this to our proximal convoluted tubule, and then from there to our Bowman's capsule. Now, again, we are talking about the movement of water and ions, water and stuff. I'm not sure where that 6 came from. And our driving force for this, as we've talked about up to this point, has been the osmolarity. So if I told you, and remember osmolarity is measured in milliosmoles. If I told you that the filtrate, when it was first produced, well, let's say it this way. If I told you that blood plasma has a osmolarity of 300 milliosmoles, then hopefully you would not be surprised to know that the filtrate, when it produced, is at 300 milliosmoles as well. And by the time it reaches the top of the loop of Henle, and over 60% of our filtrate has been reabsorbed, what do you think the osmolarity of our filtrate is at this point? 60% of it has been reabsorbed. What do you think the osmolarity is? No one wants to hazard a guess? Half, that's not a bad guess. However, remember, osmolarity is the measure of the stuff in the fluid. As stuff gets moved back into the body, remember, as we talked about, we have that obligatory water reabsorption where the water is going to follow the stuff. So what happens is the osmolarity really hasn't changed. It's still going to be 300 milliosmoles because of that obligatory water. Every time some stuff has moved back, water has followed it. And so because of that, the overall volume of our filtrate has changed. But there's my squamous cells. Not drawn to scale, but you get the idea. The concentration of stuff in the filtrate has not changed. So it has stayed pretty much the same. All right. Now, let's follow the path. Go ahead and move on to the distal convoluted tubule here of our filtrate. But I don't want to start here in the descending limb. I actually want to start here in the ascending limb and talk about what's happening over here in the ascending limb. As I mentioned, our ascending limb is impermeable to water, but we are going to be actively transporting. So of course, actively transporting means using ATP. Our solutes, and that's primarily sodium, chloride, and to a lesser extent, potassium, out of the filtrate. So what's going to be happening here, let's go ahead and use some color, is we are going to be taking sodium, we are going to be taking chloride, we are going to be taking potassium, and we are going to be moving it out of the filtrate into the interstitial fluid of the medulla, right? This is primarily happening in our, or extensively happening in our juxtamedullary nephrons. Which, remember, let's go ahead and draw a line. Between our cortex and our medulla. So we're going to be actively transporting sodium, chloride, and potassium out of the filtrate. But because it is impermeable to the water, this time the water cannot follow. So there are two main changes that are going to be taking place. First, in the tubule, what is going to happen to the number of solutes inside the tubule? Is the number of solutes going to go up, going to go down, or stay the same?
It's going to go down.
Yeah. So we have a decrease in the solutes in the filtrate. But what happens to the amount of water that's in the filtrate? Does it stay the same? Does it go up, or does it go down?
Stays the same.
Yeah. So we have the same amount of water, but less stuff in it. As a result of that, is that our osmolarity. Is our osmolarity going to stay the same? Is our osmolarity going to go up, or is our osmolarity going to go down? It's going to go down. Excellent. The net effect is that the osmolarity goes down. And in fact, by the time our filtrate reaches the end of the loop of Henle and enters into the distal convoluted tubule, the osmolarity of our filtrate is 1/3 of what it was before. So at this point, it is very dilute. Excellent. But that's what's going on in the tubule. Let's think about what is happening inside of our medulla. Now, as we are pumping solutes out of the filtrate, there are less solutes left in the filtrate, right? Again, that's a silly comment, but it kind of makes sense. So, do you think you're going to have the same amount of solutes being pumped out up here at the top of the ascending limb as you do at the bottom of the ascending limb? Same amount of solutes are going to be able to pump out, or less, or more?
Less at the top because there's less solutes in it.
Absolutely. So the second thing that's happening here, and this is what's happening, is that we are getting an uneven pumping of solutes out of the limb, where there is more pumped out at the bottom and less at the top. The net effect of that is that we get more solutes down here and less solutes up here in the interstitial fluid. And so what ends up happening is we actually produce a, I'm not going to fit this in here. I can sneak this in down here. We actually get an osmolarity gradient. Actually, I'm going to cheat and put it up here. We produce an osmolarity gradient in the medullary pyramid. Where there's more solutes at the bottom, so the net effect is an osmolarity gradient. Where again, up here in the cortex in the interstitial fluid, it is 300 milliosmoles. But starting in the medulla, it's going to start getting steeper, 400, right? 600. 800. And even by the deepest part of the medulla, it can be down to 1200 milliosmoles. Let's try to distribute that a little bit more evenly. So basically what's happening here is the unequal pumping of ions into the interstitial fluid sets up an osmolarity gradient in the medulla. Oh, let's be consistent now. More is being pumped out here at the bottom. And less at the top, producing that osmolarity gradient in the medulla of the pyramid. All right. Questions on that? So that is what's going on the ascending limb. So let's talk about how that affects the descending limb. Remember, the descending limb is our simple squamous cells that solutes aren't able to move through, but water can. So let's think about what's going to happen here. Our filtrate comes down to this part of the loop of Henle right here and has an osmolarity of 300 milliosmoles here in the tubule, but it is surrounded by interstitial fluid that has more stuff in it. The osmolarity of the medullary pyramid surrounding is at 400 milliosmoles. So what's going to happen? Well, where does water like to be?
Likes to diffuse towards the higher solute concentrations.
Yeah. Likes to go to where it's more stuff. So is it going to want to stay here in the filtrate?
Nah.
No, it is going to leave and exit into the medullary pyramid. And enough of it is going to leave where what's going to happen to my osmolarity of my, oops, that there, of my filtrate. It's going to start at 300, but by the time enough water is left, where will it stop leaving at this point? It's not a trick question. Water will continue to leave till what point is reached?
The ascending one.
Equilibrium.
Yeah, it's going to reach equilibrium. And what would the equilibrium be at this particular point in our tubule?
400.
400.
Yeah, it's going to reach 400 exactly. Right, as a result of this. Right, let's write this in the way we did the other one. Right, here in this case, what we have is right, water, not actively, it's passively, right, using osmosis, leaves the tubule, leaves the filtrate into the medullary pyramid, where there's more stuff. So water is being drawn out of the tubule. Now, notice the effect that that has on the inside. The amount of water is increasing, decreasing, or staying the same inside the tubule.
Decreasing.
Decreasing, excellent. But the amount of solutes stays the same. So we have same amount of solutes, but in less water. So essentially we're concentrating the water. And more importantly, that causes the osmolarity to increase. And as we just pointed out, it is going to increase until it reaches equilibrium with the surrounding interstitial fluid. So as our filtrate continues to flow down, by the time it reaches this point here, what is the osmolarity of our filtrate?
600.
There you go. And here?
800.
800. Excellent. And by the time it reaches the bottom, it is as concentrated as 1200 milliosmoles. Notice this is 4 times where it started. Of course, notice the flip side of this. As we come up towards the other side, now, like we've talked about, we have a ton of solutes inside of here, a massive amount of which can be pumped out and will continue to be pumped out as they work their way up. However, notice here we are using pumps to pump it out. So are we reliant on the osmolarity of the surrounding tissue to determine what the osmolarity is going to be? No, the more ATP we use, the more we're going to pump. So by the time it gets to this point, it's going to be down closer to 900. By the time it gets to here, it's going to be somewhere around 500. About here, it's going to be somewhere around 300. About here, it's going to be somewhere around 200. And by the time it leaves, as we mentioned, it is going to be all the way down to 100 milliosmoles. All right. Again, this one over here is passive, so it is reliant on the osmolarity that surrounds it. Whereas over here, it's active. And since it's active, it relies on ATP and has nothing to do with the osmolarity. The osmolarity doesn't affect or drive the process at all. Notice in this fashion, we can absorb a massive amount of ions and a large portion of the water. But we're finally able to decouple this process so that we're no longer reliant on the osmolarity to keep this process going. We can finally decouple our solutes and our water and reabsorb them separately. So let's finish the process. Water is drawn out. So again, this is water being drawn out. And over here it is sodium chloride and potassium. Excellent. Questions on this? All right, excellent. Let's go through it again.
Sorry, quick question. So on the left side, it's water being drawn out. On the right side, it's sodium chloride and potassium, correct?
Correct, yes. Over here, it is water that is being removed. And over here, it is the ions, sodium chloride and potassium, that are being removed from the filtrate. Instead of the salts going and the water following, we're able to decouple them and do them separately. And that's why we're able to change the osmolarity. Because we notice up here, we couldn't change, let's change the color of this. Up here, we couldn't change the osmolarity because of that magical thing that we talked about, obligatory water reabsorption. Everywhere the stuff went, the water had to follow. Now notice I keep saying obligatory water reabsorption, not just water reabsorption, which should be a strong dead giveaway that there is another type of water reabsorption we're going to talk about. But here, because of that obligatory water reabsorption, anytime we pulled out a solute, water followed. Here, finally, we were able to decouple those. We can bring back solutes and water separately, and that allows us to change the osmolarity. Right? There's a final implication to this that we haven't talked about yet. And why is this important that we decouple them? And the reason it's important that we decouple this is because by being able to decouple water reabsorption and solute reabsorption, we're able to change the condition of the urine, right? Which again, isn't the most important concept that we have where we change the condition of the urine, but we want to be able to change the condition of the urine based on the conditions of the body, right? If we are well hydrated, then we are fine releasing a large volume of dilute urine, right? We're totally fine with that. However, what happens if we're poorly hydrated? Do we want to lose a lot of water from our body in the form of urine?
No.
No, we want to be able to reabsorb more water. And as a result of that, we are going to have a smaller volume of more concentrated. So this ability to change the condition of the urine based on the condition of the body, to be able to hold on to more water or let the water go if it's not as vital, is one of the major important implications of this process. And notice this process relies on this concentration gradient that has been set up inside of our medullary pyramids. That's why it's so important. Nope, I wanted that to stay at purple, I guess. That is why it is so important that this occurs. I guess that can stay purple as well. Here in our juxtamedullary nephrons, because our juxtamedullary nephrons, remember, the ones that have the big, huge, long, massive loops of Henle that go down deep into the medullary pyramids. All right, I've done it here on the board, but let's look at the pretty picture from your textbooks.
I have a question about dialysis.
Yes.
Dialysis patients, they're limited on the amount of fluid that they can consume every day. So like they kind of don't pee, like they rarely pee. Is that like to just decrease the amount of work on the kidney so it's not processing as much fluid? Why can't they drink?
I don't know the answer to your question, but my guess would be that one of the reasons why it's so important to maintain the amount of water that they're ingesting is because, as we know, your blood volume affects your blood pressure. And so my guess is if they're retaining a lot of water or holding on to a lot of water, which is something they're probably doing anyway because of the problems with that, they don't want too high of a blood pressure, because my guess is that that can affect the dialysis process. So my guess is, as we've learned, pressure is very important for maintaining that glomerular filtration rate and net filtration pressure. So my guess is that that is one of the easier ways that they are able to help to regulate that. So that would be my guess. But again, it's a guess. I don't know for certain, but it's probably a fairly educated and likely correct guess. All right. Excellent. Great questions. All right, let's take a look at the pretty picture again here. So just like we did on my board, we are going to start with that thick segment of the ascending loop. That is that simple cuboidal epithelial tissue. Remember, here we are using active transporters to move things out of the tubule. Notice here we have a pump. That's what I need. So this is a pump that is using ATP, and that pump using ATP is moving chloride, sodium, and potassium all out of the tubule into the cell. Notice here we have a co-transporter. This one, interestingly enough, uses potassium, because we know potassium wants out of the cell. So we can use that driving force of potassium to take the chloride out as well. Here we have our sodium potassium ATPase, the pump, moving our sodium out of the cell. And notice potassium kind of is doing its own thing, right? Some of it is being pumped out and all the way back. Some of it is coming back in. Some of it is diffusing out from leak channels. Again, remember, one of the things we've talked about is there's a very strong driving force to reabsorb sodium and chloride, but not as much for potassium, because remember, we want relatively low levels of potassium in our blood and interstitial fluid. So as you can see, there are kind of countercurrents going on with potassium, coming in, going out, coming out, going out, and so on and so forth. So there's a small driving force of it in, but not nearly as strong as we see for the sodium and the chloride. Now, as we talked about, water cannot follow in this case. So we're able to get all these solutes out of the tubule. So they've got the same amount of water and less solutes. So as a result of that, our filtrate gets more dilute, but our medullary pyramid gets more concentrated. And remember, it's not evenly distributed either. We actually have this concentration, osmolarity gradient that is set up in the medullary pyramid, where it starts up here at 300 and can as much as be 4 times as high, 1200 milliosmoles at the depth of the pyramid. I think your book, I think I've got the picture right here. Yeah, here we go. Excellent. Notice again, here we see in the cortex, it is 300 milliosmoles throughout. It is just in the pyramids where we are able to produce this osmolarity gradient. And that osmolarity gradient is going to drive that water reabsorption in the descending limb. So again, this process does 2 things. It dilutes our filtrate, but it also sets up an unequal osmolarity gradient in our medullary pyramid. This is important because then in that thin limb, our water is going to be drawn out. It's got the simple squamous cells that are permeable to water, but impermeable to solutes. So yes, there is a strong driving force for the sodium to want to come in, but it can't. But what can happen is the water can leave. And that's exactly what's going to happen. Water is going to be drawn out of the filtrate. So the amount of solutes in the filtrate stay the same, but the volume of water changes, basically concentrating our filtrate, making it more concentrated, increasing its osmolarity. All right. Now, notice obviously the higher the osmolarity, the stronger the driving force on the water. So you would think more water would go out into that area. And if more water goes out to that area, isn't it going to dilute that area? Yeah, right? This is an issue. We don't usually get these types of gradients that set up, because what's going to happen is the water is going to want to move from the areas of the low to the areas of the high. The solutes are going to want to move from the areas of the high to the areas of the low. So how do we possibly maintain this? Well, if you remember, on those juxtamedullary nephrons, there is that very special capillary we talked about. That lattice ladder-like capillary known as the vasa recta. Now, obviously, there's a lot of active transport taking place here, so it's bringing oxygen to these cells for that, but it has its own countercurrent mechanism, which is going to help to maintain this osmolarity gradient. Your book's got this. Hold on, let's come to here, and then we'll come back to this. Notice, again, we have a countercurrent mechanism. In this case, it's what they call the countercurrent exchanger. We have blood moving in opposite directions to each other. But the big difference between the vasa recta and the loop of Henle is both limbs of the vasa recta are permeable to both water and ions. So then notice what happens as the blood travels down and up the vasa recta. As it travels down the vasa recta, it is going into deeper and deeper parts. And as it's going into deeper parts with higher levels of osmolarity, our sodium and chloride are going to want to enter into it, and water is going to want to leave. And then sodium comes in and water wants to leave. And sodium comes in and water wants to leave. So notice the effect of this is by traveling down, it takes the sodium, it takes the solutes, and brings them to the lower part of the medulla. And then the opposite is true on the other side. On the other side, I don't know if that's going to end out. Let's use different green. As it is going up, it is going to areas that have a lower osmolarity to them. So that means water is going to want to come in where there's more stuff, and the solutes are going to want to come out, and water is going to come in and solutes are going to come out, and water is going to come in and the solutes are going to come out. So notice in this case, basically on the ascending limb, it is taking the water and it brings them to a higher level. So working together, notice solutes are brought down, and water up. And this is going to help to maintain that osmolarity gradient. Where there's more solutes at the bottom, less solutes and more water at the top. So yes, normally we know things like to try to equalize, but thanks to the vasa recta, we are able to maintain these gradients and allow the loop of Henle to continue to do work. And I'm not going to try to go back to my drawing and draw the vasa recta on top of it because it's already a mess. And if I did that, it would just make it worse. But I will point out, and we'll go back to the previous slide in just a second. I believe I have the slide here. No, okay, I have it at the end. There is a nice review illustration that has all of this together on it in your textbook. All right, questions on that? So again, I think this is what we were talking about. The reabsorption of the water concentrates it 4 times that is the blood plasma, concentrates it here, and that driving force of the solutes allows them to be pulled out on the other side. So again, it just brings it back together, what we had talked about before. At the end of this process, as we mentioned, we have only 1/3 of the original volume that entered the loops of Henle. So 2/3 of the filtrate have again been reabsorbed. And it is much more dilute than it was when it entered the loop of Henle. So if we are in homeostatic balance, that filtrate stays dilute. We fine tune it in the distal convoluted tubule and in the collecting ducts, and you produce a large, and again, I say large, but again, we're talking about 1 to 2 liters in the course of the day, a large volume of dilute urine. All right. And again, we see that with this pretty picture here. Notice here we're seeing the osmolarity of our filtrate being concentrated down the loop of Henle, being diluted along the ascending loop of Henle, and then that large volume of dilute urine is released. But this only occurs if we're in homeostatic balance. What if our blood volume or our blood pressure is too low? Then, as we know, our posterior pituitary will release antidiuretic hormone. And remember, we mentioned how that antidiuretic hormone allows the distal convoluted tubule and collecting duct to express aquaporins. And that allows for the reabsorption of water. Now, obviously, the distal convoluted tubule is located up here in our cortex. And what did we say the osmolarity of the interstitial fluid in our cortex was?
100.
No, not in the filtrate, in the cortex itself. The interstitial fluid in the cortex was what?
300.
300, absolutely. It is 300 milliosmoles. So you're right. If it's 100 here inside of the distal convoluted tubule and 300 outside, and I suddenly put a bunch of aquaporins on the surface of these cells, what's going to happen to the water? It's not your
It's going to
question.
go into the cortex.
Yeah. Water is going to leave. Oops. Make this a teeny bit smaller. Water is going to leave the filtrate and go into the cortex, right? And in fact, if you think about it, we could triple the concentration of our filtrate here in the cortex, in the distal convoluted tubule, get it to 300 milliosmoles. But remember also, we talked about how antidiuretic hormone allows those principal cells in the collecting duct to be able to express aquaporins as well. And that collecting duct goes through the medulla, where we have an osmolarity gradient from 400 to 1200 milliosmoles. So if we're opening up a lot of aquaporins here inside of our collecting duct, we can pull back a lot more water, and we can make this urine very, very concentrated and a very small volume, which this pretty picture right here shows. Notice now, thanks to antidiuretic hormone, we can express those aquaporins. We can absorb much, much more water. And because of that steep concentration gradient in our medullary pyramid, notice our collecting duct actually plays a much bigger role in that concentration of the urine than our distal convoluted tubule does. Again, a perfect example how we said the collecting duct still can process the filtrate, and that is why it is still filtrate when it is in the collecting duct. It is not until it leaves the collecting duct that it's finally done being processed, and we call it urine. And so the more antidiuretic hormone we produce, the more aquaporins we express, the more concentrated the urine is going to be. Questions on that? All right, as promised, I believe it's the next slide. Oh, we'll come back to that in a second. Actually, let's do this first. Notice this type of water reabsorption is hormone dependent. Right? It's not happening all the time. Right? Only when the antidiuretic hormone tells the cells to express aquaporins. Right? That makes sense? Excellent. Silence means yes. So notice this type of water reabsorption only occurs when we get help. Right? Only when we get help in the form of that hormone helping these cells to express those aquaporins. Because of that, this type of water reabsorption we call facultative, right? Because if you facilitate something, you are helping, right? You're helping it to occur. So that facilitative reabsorption, we are facilitating, we are helping it to occur. It only occurs with antidiuretic hormone. Remember, obligatory has to occur. Water doesn't have a choice. It has to occur, and that obligatory water reabsorption occurs in the proximal convoluted tubule. Notice this type of water reabsorption occurs only in the presence of antidiuretic hormone, so only when it gets that help, and it occurs in the distal convoluted tubule and the collecting duct. And, and that kind of comparison type of condition is something that always seems to make good essay questions, right? So being able to understand the difference between obligatory and facultative water reabsorption is probably something that you should make sure you know. All right, excellent. Lastly, again, as promised, here is that pretty picture from your textbook that does a nice job of putting it all together, showing how these two countercurrent mechanisms, the countercurrent multiplier, where we are dramatically increasing and then dramatically decreasing the osmolarity of our filtrate, and the countercurrent exchanger, where we have movement of sodium and chloride into and out of the vasa recta that is going to maintain that osmolarity gradient that allows it to do work. So two countercurrent mechanisms that work together here in the medulla, in these loops of Henle of our juxtamedullary nephrons to help to produce dilute, which then can be concentrated urine. So there's that. And here's that concentration versus dilute. All right. Questions on that? What's the disagree? I missed that.
Scratch it.
Oh, okay. All right. Any other questions on what's going on the loop of Henle, or this ability to produce dilute versus concentrated urine? All right, excellent. That brings us to the distal convoluted tubule. The distal convoluted tubule, remember, as we've talked about, is where we are going to fine-tune the filtrate. And again, is it really the filtrate? Is it really the urine that we're caring about fine-tuning? No, we want to modify the conditions of the blood. based on the body's needs. And how do we tell the kidneys what the body needs? Well, how did we tell it that the body needed water?
Hormones.
Bingo. So we're going to use those hormones. Notice, again, by the time we've reached our distal convoluted tubule, we have a very dilute filtrate. It only has about 10% of the original solutes that were inside it, and only about 25% of the original water. So again, a vast majority have already been brought back. But we're going to use hormones to tell the kidneys what else we need. Right? And we've talked about many examples of this in the class. We have all of the glucocorticoids. Where were those glucocorticoids produced again?
The renal cortex.
Okay, so not the renal cortex. It wasn't a cortex.
Adrenal cortex.
Say again.
Adrenal cortex.
Excellent. And what specific layer of the adrenal cortex?
Is it the zona fasciculata?
Nope. You got the zona part right, but which zone was it? Well, now that we know it's not the fasciculata, you got a 50-50 chance.
I forgot how to say it.
Zona
Starts with A.
glomerulosa.
Yeah.
Thank God for phone a friend.
Exactly. Excellent. So yeah, in the zona glomerulosa of the adrenal cortex, we produce all the glucocorticoids. Of course, remember, as we mentioned, aldosterone is the most common of those, and they tell the kidney which electrolytes we need to be able to. I'm sorry, I said glucocorticoids. You were right. Glucocorticoids are produced in the zona fasciculata. I meant mineralo. Mineralo. It doesn't look like. There we go. Mineralo. There you go. Mineralocorticoids. I apologize. You were absolutely correct. I was wrong. Mineralocorticoids are produced in the zona glomerulosa of the adrenal cortex. They're the ones that reabsorb the electrolytes. Remember, the most common mineralocorticoid was aldosterone, telling us to reabsorb sodium, but there are ones for magnesium, there are ones for chloride, there's ones for all sorts of other electrolytes that we need as well. Chloride again as well. And again, we've also talked about how in this process, potassium is not as important, so it tends to lead a role towards that as well. Parathyroid hormone tells the kidneys to hold on to what again?
Calcium.
Calcium, excellent. So that was calcium. Antidiuretic hormone we already talked about is the fine tuning of water. So again, we've talked about lots of examples of hormones that tell the kidney things to either hold on to or let go, right? Parathyroid hormone tells it to reabsorb more calcium, whereas remember we talked about calcitonin, tells it to reabsorb less calcium. So both we can increase and decrease. We can fine tune the conditions of the blood. And really that's what we're doing. You know, we think of it in terms of fine tuning the filtrate, but really what we're doing is modifying the conditions of the blood. We are fine tuning the blood, making sure that the blood is clean and appropriate. Right? That is what we are looking for here in the distal convoluted tubule. So again, get rid of hydrogen, bring back bicarbonate, regulate the interstitial fluid. But also in the distal convoluted tubule, this is one of the areas where we are going to get rid of many of the important wastes. Some of these things, like we said, are things like neurotransmitters, hormones, Other types of chemical signals can be released this way. Toxins and wastes. Again, that pesky friend of ours, urea, is constantly sneaking in. So it's not surprising that here in the distal convoluted, one last time, we need to try to kick it out. And again, we're going to be using active and passive transport to be doing these things, getting these things in. Because the goal is anything we can get into the filtrate is going to leave the body. But remember, as we've also talked about, our filtrate is still going to be processed in the collecting duct as well. Remember, in the collecting duct, it is no longer in the nephron, but it is still filtrate. It can still be processed. And if you remember also, we mentioned that there are two main types of cells that are found in the collecting duct. The first are the principal cells, and that's what we see an example of here. Principal cells play an important role in both the reabsorption of sodium and the secretion of potassium. So again, playing a very vital role in maintaining the ionic concentration of our interstitial fluid. That facultative water reabsorption with the expression of aquaporins from our antidiuretic hormone, and our intercalated cells, which I don't think I have a picture of. Yeah, I don't. Play an important role in, so principal cells are more on osmolarity and ionic concentrations, whereas our intercalated cells are more about maintaining blood pH. Play an important role in reabsorbing bicarbonate, getting rid of hydrogen ions, and again, because that wonky potassium, we love it, we hate it, we want to move it around, it actually can help to reabsorb potassium if we're getting rid of too much of it. After all of this work, when it finally, there we go, and here's the pretty picture from your textbook. Notice in this case, we are making concentrated urine in this case, with all that fine tuning. But after all this processing, when it finally leaves the collecting duct into the minor calyx, it is finally urine, no longer processed at this point. The whole rest of the urinary system, known as the urinary tract, is just about collecting, storing, and releasing it when socially appropriate. So at the end of all this, we produce on average about 1 to 2 liters of urine during the course of the day. Mostly water, 93 to 97%, depending on conditions of your body, and nitrogen-based wastes. Again, there can be some variations in these things, but typically healthy urine is a straw yellow color, although there's some variations in the color. One of the primary pigments of your urine is urobilin. I remember that came from the breakdown of bilirubin in the making of the bile and in digestion. Typically, it should not have any type of particulates in it, right? It should be relatively clear. It is slightly more dense than water. It has a specific gravity slightly above 1. Water is, of course, 1. Again, you may never have thought of the specific gravity of urine, but maybe during the middle of the night, you got up to go to the bathroom and didn't want to wake everybody up by flushing the toilet. So when you wake up in the morning and go back in there, you see that the urine has all condensed down at the bottom of the bowl. If you've not experienced it, then I strongly encourage you to do that tonight. Pee and don't flush, and then you can enjoy that experience in the morning. It is sterile. Again, that doesn't mean you should be washing your face with it or anything like those other types of holistic things that you should be occurring. It is slightly acidic, although it can vary dramatically. If you are more of a carnivore type individual, it can become much more acidic. And for some vegans, the urine can actually become alkaline in nature. Lastly, it is slightly aromatic. Now, I know it's been a long time because we've all been locked at our houses, so it's been a long time since you've walked around downtown Sacramento. But as I walk around downtown Sacramento, slightly aromatic is not what comes to mind. However, you have to remember what we're talking about here is that the urine, when it is first produced, right? Urine is warm, it is moist, and it has a tremendous amount of biological materials, especially those nitrogen-based wastes, that bacteria love. So while it is sterile and slightly aromatic when it is first produced, as it sits around for a while and the bacteria gets in it, it certainly is no longer sterile, and it takes on that pungent aroma that we all associate with downtown Sacramento. Now, again, as it mentioned here, it varies. What kind of things can cause the smell of your urine to change?
Asparagus.
There you go. Absolutely. Asparagus is probably the classic case of that. There are also some medications that, for instance, can change the color or even change the aroma of it. As we talked about, uncontrolled diabetes mellitus can give it a sweet smell from that glucose that is in it, things along those lines. Yeah, the problem with the jellyfish. So again, when you get that jellyfish sting, there is that old wives' tale, I guess. I'm not sure what you'd call it, where if you urinate on it, it's going to be better as a result of that. When the jellyfish stings you, you actually want something more basic on it than urine. The acidity of the urine isn't something that necessarily is going to help it in any fashion. You're going to be much better off putting something more basic on it instead of the urine. So again, feel free to pee on each other if that's what gets you off, but it's not going to help with your jellyfish sting. All right. Questions on that? All right, excellent. The last thing we need to talk about, yes, we'll get to the reproductive system next. All right, so the last thing we need to talk about for this section is the urinary tract. Again, as we talked about at this point, it contains urine. Its process is just to collect, store, and ultimately the removal of the urine from our body. Let's go ahead and take our last break here, and we'll come back and work our way through the urinary tract and the processes involved in that. Looks like it is 10:13 now, so let's come back at 10:28. And I will start the recording at that point. All right. Any questions on that? All right. Excellent. See you guys in 15 minutes. So let's go ahead and finish things off. Excellent. So our last component that we need to talk about is our urinary tract. And as we talked about on the very first day, it's basically comprised of 4 main structures. Those 4 main structures are the paired ureters, the bladder, and then the urethra. So let's talk about those in part. Starting first from the ureters. Remember, the ureters are one of those retroperitoneal organs. And remember, if we're thinking of this in terms of the path of urine, we know that as it is released from the collecting duct, out the papillae of the medullary pyramid, it goes into the minor calyx. From there, it'll fall into major calyces. And I'll just stick with the singular. And then from there, remember the major calyces feed into that funnel-shaped structure, which is the renal pelvis. And that renal pelvis then feeds into the ureter, right? The renal pelvis is where it actually leaves the kidney. So essentially, this little bit we're seeing peeking out right here would be the renal pelvis. and then it feeds into those paired ureters. Now, again, we already have talked about how these are retroperitoneal. And because obviously the kidneys are above the bladder and the ureters carry the urine from, how is urine moved in the ureters?
Gravity.
So gravity would work, except what happens when you're sleeping at night? What happens if you're on the International Space Station for 10 months, right? Do you not urinate the whole time you're up there? So while it certainly gravity can help to facilitate the process, of course, the primary way we're going to use it is indeed peristalsis. Right? So that way you are still able to remove and move the urine from the kidneys, no matter what your position is, whether you're laying down in bed, whether you're doing a handstand, like I said, whether you're in outer space where there's no gravity. All of those are important times where we still need to be able to move our urine out of the kidney. Remember, we want to keep that, we don't want that urine backing up into the kidney. That increase in pressure can cause a lot of problems. Now, one of the interesting things, you don't quite see it from this illustration, but if we were to rotate this individual, this female, to the side, one of the things that you would notice is that the ureters actually come behind the posterior part of the bladder and actually insert into the bladder from an inferior and posterior position. So they're not attaching to the top up here, they're actually going behind it and connecting down below. Why might that be useful?
When giving birth, maybe.
True. It could be because it's given birth. Remember, also it's retroperitoneal, so it could be useful that way. What else? I like keeping them out of the way for birth. That's definitely an important thing. But remember, there's two other issues as well. First is that as the as the bladder fills with urine, it is going to expand. And if you think about it, if the ureters just connected to the top, as it expanded, that could actually cause a kinking in the ureter. And remember, as we talked about, that would be a very bad thing because we'd get a backup of pressure as a result of it. But remember also, the bladder has some elasticity in it. Not as much as it could, because thankfully it is lined with that transitional epithelial tissue. But still, as it expands, there is going to be an increase in pressure here inside of it. And we don't want that pressure to back up into the kidney. So notice as the bladder fills and gets larger, it is actually going to push down further and harder on that opening, that os of the ureter, and help to keep it closed. All right, so as you're squeezing it from the top, putting that pressure on it from the top, it's going to help to keep it closed. Now, if it's being forced closed by the urine, how is it going to get the urine out of the ureter and into the bladder to begin with? Well, didn't we just answer that question? Peristalsis. That peristalsis, those rhythmic contractions of the ureter, will help to force the urine into the bladder. And then when it's not contracting, it'll close again to maintain the pressure. So basically, it works as a pressure valve. It allows for the expansion of the bladder without any kind of worrying about kinking the line, and it acts as a pressure valve to make sure that the urine doesn't back up into the kidney as well. Now, one of the challenges, both in a good and a bad thing, if you remember, one of the things that we've talked about is that the entire urinary tract, is lined with what type of epithelial tissue?
Transitional.
Yep. With that transitional epithelial tissue, which is a good thing. It makes it very easy to identify the urinary tract. The problem is that it can make it tricky then to tell the three organs apart. How do you tell the ureter from the bladder from the urethra if they're all aligned with a transitional epithelial tissue? And the key to that is going to be to focus on some of the main other characteristics. So let's look at some histology for this. Here we see the histology of the ureter. As I'm sure you remember from 430, as you look at this ureter, you will indeed see it is a stratified tissue comprised of many layers of cells. Notice one of the key characteristics to our transitional epithelial tissue is that the cells typically get bigger from the basal surface to the apical surface. And notice on the apical surface, we have these big block-shaped cells. I haven't looked closely to see if those are the case in this one. Ah, there we go. There was one other, if you remember back to 430, there was one dead giveaway that you were looking at a transitional epithelial tissue. Does anybody remember what that dead giveaway that you were looking at a transitional epithelial tissue was? It's okay if you don't, it's been a while. If you remember, on those big block apical cells, occasionally, not in every one, but every once in a while, you can find a cell that has two nuclei. If you see a cell that has two nuclei, and it looks like we have a couple of them. This is a low magnification, so it's a little tricky, but it looks like we probably have one there, and it looks like we probably have one there. And maybe when we get a closer look at this transitional tissue, you can see that. But occasionally, it doesn't always happen. You're not always going to see it in every plane of section. But when you see that, there you go, multinucleate. When you see that, that is a dead giveaway. You are looking at a transitional epithelial tissue. So that makes it really, really nicely there. Of course,
But not necessarily the ureter, just a transitional epithelial.
correct. This is true for all transitional epithelial tissues. Absolutely. So again, it's good because transitional is pretty easy tissue to easily identify. So we know we're in the reproductive tract, I mean the urinary tract, but you're right. Part of the problem with that is it's in all four organs. So identifying a transitional epithelial tissue is easier. Telling the four organs apart is a little bit more challenging. Really three. Obviously, you're not going to be able to histologically tell the difference between the right ureter and the left ureter. So let's talk about other things we know. Obviously, this is an organ that is open to the outside world. So what type of membrane lines it?
Stratified columnar epithelial tissue.
Well, but not, no, so remember, we know it's a transitional epithelial tissue. There we go, a mucous membrane. Remember, mucous membranes are the ones that are open to the outside world, which of course tells us that while we have that transitional epithelial tissue on top, we also know all mucous membranes have what type of connective tissue that helps to form that membrane?
Areolar connective tissue.
There we go. And what do we call it based on its location?
Lamina propria.
There we go. Excellent. Notice with our ureter, we have a relatively small lamina propria. And notice with this small lamina propria, even though it's a mucous membrane, do we see a large number of really big mucous glands? No, because this isn't an organ that is going to produce a lot of mucus. We certainly don't need mucus to be able to, grease the wheels for urine to be able to move through it. Obviously, we need to produce peristalsis. So notice we do have a layer of smooth muscle, right? Unlike the digestive system, there are not two layers, just a single circular layer to it. And again, notice because we know the ureter is retroperitoneal, it is surrounded by that adventitia. Notice there's some adipose and some other connective tissues that are going to be holding it in place. Last thing, and I know it can be a little bit tricky, but obviously the ureters are going to be the smallest of these urinary tract organs. Right? This one happens to be a human one, but often the ones you'll look at on the microscopes and stuff like that will be from mice and things along those lines. And they're incredibly tiny, but even a human ureter is small enough we're able to take a slice of it and put it on a single slide and being able to see that. Are you going to be able to take a slice through the entire circumference of a bladder, heck, even a rat's bladder, and get that whole bladder on a single slide?
Not with that attitude.
Exactly. Not with that attitude. Exactly. Right. You need to bend the laws of physics to be able to do it, but you've absolutely got the right idea. So no. So that's, again, these are the types of things you're going to look for. It is going to be tricky because the ureter histologically is very similar to the urethra. But the size, the lack of the mucous glands, and those are the things that can kind of help you to be able to distinguish it. Luckily, the bladder is going to be very different, so that won't be too scary. But we'll see, this is similar to the urethra, so we'll have to take some tricks to try to figure them apart. All right, let's talk about that bladder. It is a big, smooth, collapsible sac, just like the stomach. It has rugae, just like the stomach. And just like the stomach, these allow for the expansion of that organ with, again, a decrease in pressure, right? When you take a paper bag and you crumple it into a ball and then you open it up, right? You're just unfolding it. You're not stretching it out or anything, so you're not increasing that pressure with it. So that is a huge advantage of that. Also, like the stomach, it has 3 layers of smooth muscle. Now remember, with the stomach, there were 3 distinct layers: the oblique, the circular, and the longitudinal, right? Very distinct layers. The same is not true for the bladder. These are not distinct layers. There is lots of it, but they're kind of interwoven and intermixed with each other, so you're not going to be able to tell them apart. And because they're not distinct layers, basically they are just collectively, oops, there. They're just collectively referred to as the Detrucer muscles. So notice here we've got this simple illustration. We see all sorts of bundles, all sorts of fascicles. We can't tell which one's circular, which one's oblique, which one's longitudinal or anything like that. Just the whole thing collectively is just referred to as the Detrucer muscle. So we don't have to worry about, so we need to know that there's 3 layers to it, but we're not going to be able to distinguish them. There are 3 openings to our urinary bladder. And we see those nicely right here. Notice we've already mentioned how there are going to be, no, let's not do that. Let's do this instead. There are going to be the 2 osses for the urethra, or the urethral openings, where the 2 ureters feed into the 1 bladder. But we also have the singular urethra, so we have the internal os, of the urethra as well. And make a point of emphasizing this because not only are there 2 ways in and only 1 way out, just like the Thunderdome, but notice also that there is this thickened region of the bladder kind of forming a triangle between them here in the bladder. And this thickened funnel-shaped region is what is known as the trigone. The trigone is important for two reasons. Being thickened and hardened, it keeps the bladder from collapsing completely, helping to keep the pathway open. However, from a clinical standpoint, if it is a thickened, hardened region and we can't fully collapse the bladder, are we likely to be able to fully empty the bladder?
No.
No, exactly. And so from a clinical standpoint, the urine, there is a small amount of urine that typically remains in the bladder after expulsion. And as we've talked about, urine is warm, it is moist, and has a large amount of nutrients, metabolic nutrients anyway, for bacteria. So so when an individual gets a urinary tract or a bladder infection, it often starts here in the trigone region, because that is where you can get an accumulation of urine that is not able to be released. Now, as I mentioned, and let's clear my picture here for a second, this big, huge, thick, interlaced, three-layered structure is known as the Detrucer muscle, right? But within that Detrucer muscle, there is an enlargement of, that's not a good color. There is an enlargement of a circular layer of the Detrucer muscle right around the opening of the urethra, known as the internal urethral sphincter. Again, this is an enlargement of the smooth muscle, of the circular smooth muscle layer. And it's going to play an important role in the expulsion of urine. What does that sound an awful lot like?
Anus?
Yeah, defecation, right? In defecation, remember, in our anus, we had an enlargement of the smooth muscle layer, forming a valve-like structure. Being smooth muscle, this is going to be involuntarily controlled. And is going to play a role in the voiding of urine. In fact, the defecation reflex and the micturition reflex are incredibly similar. So the good news is, if you learned the defecation reflex, even if some of you may have had it, or not may, did have it as an essay question on your digestive exam, well, the process is going to be very, very similar to what we see in the urinary system with micturition, the voiding of urine. Excellent. So we have that circular structure there as well. Notice when we look at this histologically, again, look at the huge difference of the scale of this. We can barely make out the transitional epithelial tissue here on the surface. We can see all of these invaginations of the rugae as it is coming down and folding in on itself, allowing it to be able to collapse. Notice here in the lamina propria, we can see more glands. We didn't really see any mucus glands in our ureter, but they are here in the bladder. And again, in this case, the mucus isn't necessarily there for, again, the removal of the urine or protection from the urine. It's really more about, this is a structure that is close to the outside world, so this mucus is helping to protect us from pathogens, capturing any pathogens and dust and debris and allowing it to be removed with the urine. And clearly the most distinct feature is that big, large, big interlaced bundles of smooth muscle, that Detrucer muscle. Lastly, of course, our bladder is a intraperitoneal organ. So as such, it is surrounded by a visceral peritoneum on the outer surface, which again, of course, if we looked at up close, would be a simple squamous epithelial tissue with a tiny bit of areolar connective tissue. And of course, the simple squamous we would call a mesothelium based on its location. So again, the mere scope of it and the massive size of the smooth muscle wall to it is usually a pretty strong indicator that you are looking at the bladder. So that one's pretty easy to tell. That brings us to the urethra. The urethra, again, is a muscular tube with two openings, an internal os, which is located in the bladder, and of course the external os, which is where it is expulsed from the body. Like the ureters, it is going to carry urine out of the body via peristalsis. Now, when you first start to void urine from your body, a process called micturition, the flow is fairly consistent and continuous. And that is again because of the pressure inside of the bladder as that elasticity of the bladder is pushing on the urine and forcing it out. However, does the flow of the urine stay continuous until it stops? Do you just urinate and then stop? No. Near the end of the process, it becomes more pulsatile in nature. And there, that pulsatile nature is where you see the action of that peristalsis from the contractions of that urethra in that voiding of the urine. Now, as I mentioned, it can be tricky to tell the difference between the urethra and the ureter. Both are typically small enough to be seen on a microscope in its entirety, especially if you are looking at a comparative urethra, like in a rat or something along those lines, which are typically what you see on slides. But notice again, based on the size of the cells, we can tell this is bigger. Notice to get all of it in the field of view here, the overall size of the transitional cells are much, much smaller. Notice also that in the orifice, in the opening or lumen of the organ, there are more invaginations. To the urethra. There's more expansion of this urethra that can occur. So we see much more of these folded invaginations in the urethra than we do in the ureter. Notice it has a much larger lamina propria, or thicker. And notice in here, you are going to see much more of those, oops, let's do it this way, mucous glands. Again, this is open to the outside world, so we need that protection in the form of mucus. So there's much more mucous glands, and that can be found in that thicker lamina propria, as we're here as well. All right, so again, all of these are ways that we can hopefully fairly easily distinguish the urethra from the ureter. Size is a big one, but size can also be subjective. So we need to focus on other contents. And really, it's the lamina propria and those invaginations of the lumen that are really the things that make it easy to distinguish. And obviously notice that is a much larger smooth muscle layer as well. A lot of elasticity. The urethra actually has the ability to expand a lot to be able to accommodate. And like I said, many, many mucous glands. We did all of that. Now, as I also mentioned, here in the urethra is where we're going to see our first significant and meaningful sex differences. Right? Yes, there are other sex differences we have seen along the way, whether it was the amount of our glomerular filtration rate, whether it is the size of the brain, whether it is the height, all of those types of things are things that we've talked about in the past, that because of the just sheer numbers of individuals on the planet, small changes are mathematically significant. right? Like height, for instance, but they're not meaningful. They don't affect function or anything along those lines. This is the first time we really see meaningful sex differences in the organs that we're talking about that influence what they do, right? Obviously, one of the big differences is length. In females, the length of the urethra is only about 1 inch in length, whereas in males, it is 8 inches. Notice I'm talking about urethral length. I'm not talking about the length of your penis, right? We're talking about the length of the urethra. Obviously, one is much longer than the other. However, one of the implications that we don't really think about in terms of this is notice in the females, the bladder is much closer. Oh, how do I spell bladder really? As such, females, especially when they're adolescent and teenagers, are much more likely to get urinary tract and bladder infections than males are, because that bladder with that trigone, with that urine inside, is much closer to the outside world. So a harmful pathogen or a bacteria has a shorter distance to travel to get to that urine in the bladder in a female than it does in a male. So especially during youth, bladder infections much more common in females than they are in males. Obviously, the locations are different. In females, the urethra is located adjacent to the vagina. If we think of this in terms of the vaginal canal, then the urethra basically is anterior to it, having its own separate orifice and opening. So obviously for a female, the reproductive tracts and the urinary tract are separate. Is the same thing true for males? No. In males, basically, the urethra passes through the prostate and the penis. So from a functional standpoint, in females, the urethra only carries urine, whereas in males, it is a conduit for both urine and semen. So from a functional standpoint, the urethra in females in the urinary system only. Whereas in males, it is an organ of both the urinary system. Do it again. And the reproductive system. And so we'll talk about some of the implications of that when we actually get to the reproductive system as well. Lastly, there are some big anatomical differences as well. Again, here we see the urethra and the end of our urinary tract in a female. Again, as we've talked about, we've got that large detrucer muscle, which, let's not forget, has that internal urethral sphincter within it. So that enlargement of the circular muscle that is located right here. The urethra has to channel through and out of the body. And along that way, it has to pass through the levator ani, right? Our urogenital diaphragm, that muscular diaphragm that helps to support and retract the anus that we talked about in defecation. The levator ani muscle group made of the iliococcygeus and pubococcygeus. And if you remember from digestion, this is obviously a skeletal muscle. And just like in defecation, there is an enlargement of that levator ani, that enlargement of that skeletal muscle that surrounds the urethra, forming the external urethral sphincter. So just like we saw in defecation, we have two sphincters, an internal urethral sphincter, which is comprised of smooth muscle, and an external urethral sphincter made of skeletal muscle. Now, notice, unlike defecation, where the smooth muscle of the internal anal sphincter is surrounded by the external anal sphincter, notice here they're separate from each other. The internal urethral sphincter is part of the bladder, and that external urethral sphincter is part of the urogenital diaphragm, part of the levator ani muscle. But aside from the displacement of their locations, their functions are essentially going to be the same. And that's it. Pretty simple and straightforward for the females. Let's take a look at the male variety of this. Again, we still have the same basic anatomy. We have the two ureteral orifices or osses, the internal urethral os, and the urethra that exits the body. Again, we are going to have enlargements of the bladder, that detrucer muscle, the circular layer, forming the internal urethral sphincter. And notice again, the urethra has to pass through the urogenital diaphragm, the levator ani muscle, and as it passes through the levator ani muscle, there is an enlarged circular area. Not trying to color that gland, but it's okay if we do for right now. That enlargement of the skeletal muscle, that is the external urethral sphincter, which again, for consistency, is skeletal muscle. It is part of the levator ani, whereas the internal urethral sphincter is smooth muscle, part of the detrucer muscle. But notice the urethra has to pass through some other structures as well. And so that we divide the urethra into three distinct regions based on what it passes through. The portion, the region of the organ that passes through the prostate is the prostatic urethra. The prostate is like a donut that surrounds the base of the bladder and completely surrounds the urethra. Right? You can't watch a sporting event these days without hearing about prostate enlargement, right? And again, as you can see, because it's this circular structure, as it squeezes, as it enlarges, it squeezes the urethra, doing two things. It irritates the lining of the urethra, giving you the sensation that you need to void and go to the bathroom, and it constricts the passageways, so it's harder to void the urine, right? So that's not a very fun experience to have, but I'm sure people cope with it. There is the portion, the very, very small region of the urethra that passes through the urogenital diaphragm, that passes through the external urethral sphincter, known as the membranous urethra. I don't have a good explanation for why it's called that, but it is. And lastly, there is the portion of the urethra that passes through the penis. As such, it is known as the penile urethra. And also because it passes through the spongy tissue of the urethra, it is also known as the spongy urethra. So both of those terms, spongy urethra or penile urethra, are acceptable terms. In fact, sometimes it's also referred to as the cavernous urethra. So any of those appropriate anatomical terms are correct. So one organ, the urethra, but three distinct regions to it: prostatic, membranous, and spongy. All right. Questions on that? Excellent. All righty. did that. Oh, and here's some more pretty pictures. Notice here's the male from a sagittal view. Again, there's a couple things that we can see. Again, notice here, we can see how the ureter is coming down and entering into the posterior and inferior part of that. Notice we can see, again, the prosthetic urethra, the membranous urethra going through our urogenital diaphragm, and then the spongy urethra as it passes through those urinary organs, where we can also see it's related to the reproductive organs that we'll learn about starting next week. And to give the female here fair due, here we see again the separation of the urinary system with its own orifice and entering into the bladder, and the vaginal canal, which obviously leads up into the uterus, right? So we see those are separate openings for the urinary system and reproductive system, where it's shared piping in the male. But notice again, it passes through the urogenital diaphragm. So we have our external urethral sphincter down here, and of course our internal is going to be up here. So while they're in slightly different locations, their functions are going to be the same, which of course is the last thing we have to talk about, our Maturition reflex. And like I said, this is going to be incredibly similar to what we saw in the defecation reflex. We have an organ. So let's go ahead and make our bladder. Connect it to the urethra. And remind ourselves that we have two sets of valves that are going to play a role in controlling the movement. The first of those, which we have now done in red, is of course our, what do I want to do this? Let's do this over here. Internal urethral sphincter, which of course is smooth muscle. So of course that means it is going to be involuntarily controlled. We also have within our levator ani muscle, that enlargement, that is the external urethral sphincter, which is skeletal muscle, and it has voluntary control. Let's go ahead and move those. This can go here. This can go here. And again, I think I said it, but let's go ahead and do it. This is part of the detrucer. Much like we saw in the rectum, we are going to have stretch receptors. The walls of the bladder. And not surprisingly, this Maturition reflex is going to involve both intrinsic and extrinsic controls. So, step one, bladder fills with urine. And of course, as that bladder fills with urine, our bladder stretches. Once it reaches a critical point, then at that point, our stretch receptors are going to send signals. And those signals go two ways, two places. We have a urinary plexus where we're going to have our intrinsic control. And that intrinsic control is basically going to do two things. The first thing that it is going to do is that it is going to cause contractions to the detrusor muscle. And the second thing it is going to do is it is going to relax the internal urethral sphincter. So we get these contractions that are occurring. Let's not use that. Contractions of the bladder, obviously increasing the pressure. As we increase pressure, we relax that internal urethral sphincter, and urine is going to enter into the urethra. We have sense receptors in there. So we perceive the contraction of the bladder, we perceive the urine in the urethra, and we get the sensation that you need to void. All right? This is when you start doing the pee-pee dance. However, are you instantly voiding urine when this occurs? Hopefully not. And the reason for that is that those stretch receptors also send a signal via the sacral spinal cord to via the parasympathetic, let's say it this way. Also send a signal to the parasympathetic nervous system via the sacral spinal cord. And this parasympathetic reflex triggers the contraction of the external urethral sphincter. So you get the sensation of void that you need to void, but voiding does not occur. So what, of course, is the last step of this process? You get yourself to a socially appropriate location. Right behind a bush or wherever it is. You then voluntarily relax the external urethral sphincter. And when you do that, when both sphincters are open, voiding occurs. And that is kind of key, because what if you start feeling that sensation that you have to void when you're in the car and you just passed a sign that said the next rest stop is 30 miles? All right, you have a choice. You can stop on the side of the road and find a bush, or you can hold it and keep going. And just like defecation, if you do not void, the signal will habituate. and it goes away, right? When it goes away, that internal urethral sphincter closes. So if you don't time it right, by the time you get to the restroom, you may have trouble going. Or, and I'll be honest, I don't know if this is a problem that females have. Do females have what we call a shy bladder? Right? I don't know if it works. I don't know if it works quite the same way. But part of it is, you know, like males, you go to some sporting event, like the old Oakland Coliseum was great for this. The Oakland Coliseum, basically the male urinals were basically a big tub in the center that basically everybody stood around. It was like a communal urinal. And some people can get a little nervous about that. And as we talked about, nervousness involves a sympathetic nervous system response. And when our sympathetic nervous system responds, it actually causes a constriction of that internal urethral sphincter. And that can, you know, that can make voiding more challenging when that occurs, right? Both have to be open for that to occur. So the reason that some people have that problem with the shy bladder is because of that anxiousness, that nervousness, and that sympathetic control, which causes that constriction and makes it hard for us. Because again, even though you want to void, this is smooth muscle you can't voluntarily control. So as much as you want to let it out, it can be very hard sometimes to get that to relax. Not after having kids. Awesome. Excellent. Right? Of course, the other last point that I want to make is while it habituates, it continues to fill. And as it continues to fill, it stretches more. And as it stretches more, it produces more pressure. And as it stretches more and produces more pressure, the contractions of the bladder become stronger. So as a result of that, if you continue to hold it and continue to hold it and continue to hold it, what can end up happening?
You have an accident.
Yeah, what can happen is the pressure can become great enough, not stronger. The pressure in the bladder can become great enough to force the external urethral sphincter open. And as a result of that, you are voiding urine whether you want to or not. However, especially since many of you are thinking of going into the nursing field, another important thing to remember involves that habituation. Continuous habituation of this reflex can actually cause this reflex to fail. This is a problem, thankfully, that was seen many years ago and is not so much of an issue now, but in nurses in particular, because nurses, before they had better unions, before they had, you know, more control over their schedules and things along those lines, would work 10, 12, 14-hour shifts. And sometimes during that period of time, they would never get a break, because they're constantly running from putting out one fire here and one fire there and one fire here and take care of this patient, this patient, this patient. And there have been situations where nurses in particular, because they're constantly having to hold their urine, their urinary reflex basically habituates to the point where the habituation becomes permanent. Your internal urethral sphincter no longer responds to the stretch of the bladder. So then how do these women have to void the urine from their body? Once a day or more, they have to insert a catheter to be able to void the urine from their bladder, because they're no longer able to do it voluntarily. All right, excellent. So we did this here. Let's look at the pretty pictures. Again, bladder fills and stimulates the stretch receptors. Sacrospinal cord reflex, regulated by the parasympathetic nervous system. And using both intrinsic and extrinsic. Controls, we relax the internal urethral sphincter, we contract the external urethral sphincter so that you say the sense that you need to void, but voiding isn't going to occur. And again, remember, oh, let's put this here as well. This is also intrinsic, the contraction of the detrucer, but we voluntarily relax the external sphincter and voiding occurs. Like I said, if the volume increases and it stretches enough, usually somewhere around 400 milliliters, the contractions can become powerful enough to force the sphincter open. And I think, no, I guess not. I thought your book had a picture, but apparently I don't have it here. Excellent. With that, we have now filtered the blood. We have voided our urine. That is now everything you are responsible for on this exam. All right. Questions on that? All right. If we are comfortable with micturition, and again, it is, can't pee and swim. No, yeah, well, yes. If you are swimming, you're using your sympathetic nervous system, and so it's more likely harder to relax that sphincter. Absolutely. Although if you're in a lake, then peeing can help to keep you warm, so that is important. Excellent. All right. And it's okay to do it in your pool too. That's why you put chlorine in there, right?
But my kids can pee and swim, and they make fun of me that I can't. I have to hold on to the boat.
No? Interesting. Maybe you just have more control. You have more strength in your urethra than they do. Exactly. All right. Excellent. So that is everything we have to do. Again, all the new material that we have to cover, but I know we've covered a lot of material over these two sections between the respiratory system and the urinary system. So what I would like to do with the time that is left is do a question and answer review. Again, we'll take a quick brief break, giving people an opportunity to stretch, micturate if they need to, get something to drink, get a little bit more caffeine, and then come back with questions. Again, reviews are not me telling you what I think is important. That is what I do every day here in class. This is an opportunity for you to ask questions about the things that you are unclear about, so we can make sure that you understand them and you are successful on the exam. All right. Normally I make the threat here that, again, if you don't ask questions, I assume you've understood the material and I make the test harder, but I don't make the tests. They're randomly selected to you. I just put the questions in there, and the hard questions are already in, so there's no sense in that. So I have no control, but it's still important to ask the questions because, again, I get paid the same whether you get A's or F's, but I like it a lot more when you get A's. So let me help you to be successful in the class. If you're confused by something, ask me those questions so we can come up with the right answers and I can help you to be successful. All right, let's take just a quick 10-minute break, come back at 11:35. That'll give us an hour to ask questions and answer questions. And so meet me back here at 11:35. All right, see you in 10 minutes. So I'm now here for you. What questions can I answer to help you to be successful? What kind of stuff that we've covered over the past 3 weeks is confusing to you that I can help you with? Here we go. How exactly would you ask questions on the pulmonary circulation? Okay, so you've kind of got a couple questions in there together. So obviously the pulmonary circulation, we need to know what components from the heart, what blood vessels are involved in both the pulmonary circulation and the bronchial circulation. However, when you're talking about the partial pressures, remember the partial pressures involve both the pulmonary circulation and the systemic circulation because it involves both internal and external gas exchange. I think the simplest way to do that, like I said, you have to know those values. So I could just simply ask you, what is the partial pressure of oxygen in a pulmonary capillary when it enters the pulmonary capillary? What is the partial pressure of oxygen as in the, you know, in the blood and the pulmonary capillary as the blood leaves the capillary? Something along those lines. Or what is the pressure in a systemic artery versus a systemic vein, or something along those lines? So absolutely, those are the types of questions that I could ask, where I could just flat out ask you the numbers. Or, you know, what is the, you know, what are the partial pressures of oxygen involved in internal gas exchange, right? What are they, you know, in the tissue? What are they in the blood? And which way does the oxygen move because of it? Or, and again, we could do the same thing with CO2. So absolutely, those are values that you need to understand and how they work and why those numbers are what they are. So there's numerous ways that I could ask that, but absolutely know those numbers. All right, what else?
I'm having a hard time seeing the difference in the collecting ducts and the loop of Henle's and the proximal and distal tubules in the histology. They kind of all look the same to me.
Okay, that is, I can see that. Let's do this. I don't care about any of this. Okay, this one will work. It's not the best example, but it's the quickest one that I found. So let's use this. So I when all else fails, the first place I usually go is that CRC site, because that's usually got some decent stuff, and I have it bookmarked, so it makes it easy to get to. So identify the organ in the field of view.
Kidney,
Kidney, excellent. How do you know?
the renal tubule or renal corpuscle.
Renal corpuscle, absolutely. And so also the renal corpuscle tells us we're in what specific part of the kidney. What region of the kidney are we in?
The cortex.
Cortex, because only in the cortex are we going to find the renal corpuscle. Also in the cortex is where we're going to find the proximal convoluted tubule and the distal convoluted tubule. All right, so we're going to obviously find the renal corpuscle and all the things associated with that. But we're also going to find the proximal convoluted tubule and the distal convoluted tubule. Now, here in the cortex, how are we going to tell these two apart? What's some of the things that we could use, or what's the primary thing we can use to distinguish these two from each other? What are some of the differences between the proximal convoluted tubule and distal convoluted tubule? Obviously, they're both cuboidal cells, so that doesn't help us. They're different in the number of mitochondria they have, but we can't see mitochondria in this picture. So what can we use to tell them apart? There we go. Allison's got it. The microvilli. Which one has more?
The proximal.
Proximal. So notice the key to being able to tell these two things apart is to look at them in terms of the lumen. Notice the lumen of this piece of a tubule is very dark. There's a lot of stuff in it, very dirty appearance. This one looks very dirty as well, although I'm not sure what's going on here with this weird cut, but definitely the part of the lumen that we can see there, it's not very clear. It's very dirty. There's a lot of stuff. Here's another one that's like that as well. Compare those, for instance, to this one right here, or this one right here. Notice how clear the lumens are, right? Again, this is kind of a weird section of one. I probably wouldn't use this one on an exam, but again, you can see there's a lumen here that is much more clear, right? This one here, again, much more stuff inside of it. So really the key ways to tell the difference between the proximal convoluted tubule and the distal convoluted tubule is going to be the condition of the lumen. If the lumen is very clear, it is very likely to be the distal convoluted tubule. If it is very dirty, it is likely, or, you know, I don't know about a better word than dirty, but it looks like it's got a lot of stuff in it, then it is going to be the proximal convoluted tubule. Now, there is one other dead giveaway on this one. If you look closely right here, you can see some of the tightly packed cells of the macula densa that happen to be right next to our renal corpuscle. So obviously when you see a macula densa, that obviously is going to be a distal convoluted tubule. But this one doesn't have a macula densa, and yet we can still see the clearness of the lumen to help to distinguish it that way. Notice we're not seeing the loops of Henle, and we're not seeing the distal convoluted, the collecting duct in the cortex. Those are things that you're not going to see. It's just going to be these you're going to be responsible for. Now, if we, instead of going to this side, because I think I posted it, let's go back to our modules. I remember under our modules, oops, hold on. I wanted to do this, do this. Excellent. Does that make it bigger so you guys can see it bigger? Okay, perfect. Here we are seeing two things. We are seeing a large number of tubules that are fairly parallel to each other. And we're not seeing any renal corpuscles. So those two things right there tell us that we are in the medulla. We're not in the cortex. Okay? Now, that means that here, remember, we're not going to see any proximal convoluted tubule. We're not going to see any distal convoluted tubules. We're just going to see the loops of Henle and the collecting ducts. Now, histologically, is it possible to tell the difference between the simple squamous descending limb, the simple cuboidal ascending limb, and the collecting ducts? Is it possible to do that, do you think, histologically? Yes, but as you've looked at your histology list, are those things that you're going to necessarily be responsible for? No, not histologically. On some of the models that we've looked at and seen, you're absolutely going to need to be able to tell those apart, but not here. Histologically, though, you are responsible for the vasa rectas, those blood vessels that we can see as you see on this illustration here. For something like the histological responsibility for those things, I'm much more likely, and again, if we go back to our virtual anatomy lab for Cosumnes River College, urinary models and charts. Nah, this one's better. Something like this here. Here, can I hold you responsible for all of the components of the nephron, both the blood vessels and the tubular structures on a model like this? Yeah, absolutely. So again, if you're looking at this, and I'll just use some small arrows, whether we are doing the blood vessels from, again, oh, that doesn't show up good at all. There's our afferent arteriole feeding into our glomerulus, feeding out into, well, here it's easier to see, an efferent arteriole into the paratubular capillaries, right? So we can see those things there. Notice we also have a vasa recta represented by these blood vessels here. We also have all the tubular components. Bowman's capsule, very easy to see. Proximal convoluted tubule, distal convoluted tubule, right? A thin descending limb, thick ascending limb. And how do we know this is the descending limb?
It comes off of the proximal.
Yeah,
Convoluted
exactly. It comes off the proximal, absolutely. And then that distal feeds into our collecting duct, which travels all the way to the apex of our medulla, releasing our urine. All right, we can see the arcuate arteries and veins that help to separate the granular cortex from the striated medulla. Right, so stuff like this, we can see all of these things that we talked about. So yes, you need to be able to recognize the thin and the thick ascending and descending limbs of the loop of Henle on something like that, but I'm not going to make you do it under the microscope. All right, so this technically is histology because again, these are things we would see under the microscope, but I'm not going to make you distinguish them under the microscope. Technically, it's possible to, but I'm not expecting you to do that at this level. But on a model like this or a model similar to that, should you be able to follow the pieces and figure out what's going on where? Absolutely. All right, did that answer your question? I guess I'll take the silence as a yes.
Sorry.
No,
Yeah,
worries.
it does. I guess I was looking at some of the Yale histology slides, and the collecting ducts in like a cross section look just like the proximal and the distal.
Right, because they are, cuboidal cells, tubular things, and against the principal cells have microvilli, the intercalated cells don't. So yes, there are some similarities, but again, I'm not holding you for the histology on those under a microscope. On a histological model like this, yes, absolutely. Right? So, yeah. But yes, that is an easy rabbit hole to go down. Obviously, we could do far more with this than we are, but this is the level that I'm expecting you for this exam.
Okay.
All right. Next question.
The process of reciprocal inhibition. I have my pretty picture drawn. I'm having trouble sorting out like sequentially the process.
Okay. So instead of drawing the picture and focusing on the picture, let's just talk about the process. All right. So here's the couple key components. There are two neural clusters, two nuclei. Two clusters of cell bodies, neural cell bodies that are involved in the process. They are the, let's do it this way. Inspiratory center. And when it fires action potentials, what does the inspiratory center do?
It excites the diaphragm and inhibits the expiratory center.
True. Well, we'll get to that in a second. So you've got the right idea. It excites the diaphragm, and as a result of that, it contracts, and we breathe in. Right? We breathe in as a result of that. Absolutely. So you are correct. There is a second thing it does as well. It also inhibits the expiratory center. Our expiratory center basically has one effect. When it fires action potentials, it inhibits the inspiratory center. Right? And of course, when the inspiratory center is not firing action potentials, the diaphragm relaxes and we breathe out. Okay? So these are the two centers involved, and these are what these two centers do. So notice the key to this, the fact that they inhibit each other. Both cannot fire at the same time. because if one is firing, it's inhibiting the other. Right? Does that make sense? Excellent. And so that's good. Anytime we're going to have a circuit, we don't want them firing at the same time, right? When you are walking, you don't want the flexors in both legs working at the same time, because then both knees are going to flex and you're going to fall to the ground. You want to make sure one is firing and then the other. You want to have this circuit, this cycle where it's going back and forth, right? The problem is if one inhibits the other, and it starts firing and inhibiting the other one, it could conceivably inhibit it forever. And then you don't get the going back and forth of the circuit and either way. So there is another important characteristic to both these centers that allow them to be able to oscillate. That characteristic, well, actually, there's really two characteristics. The first is that they habituate. So when the expiratory is inhibiting the inspiratory, eventually it loses its effect. So these centers habituate to the inhibition, which means that the inhibition cannot last forever. But just having that inhibition removed isn't enough. We need it to then fire an action potential after it's been inhibited. And that's where the second characteristic comes in. It's what we call a rebound. Basically what that rebound is, normally when we talked about neurons, for instance, when we talked about a cell, well, let's actually draw this. Right, this is something we talked about back in the nervous system. When you have a cell and you hyperpolarize that cell, it stays hyperpolarized, and then we talk about how it repolarizes and comes back to rest. Right? Does that sound vaguely familiar? We talked about this a little way back in 430. You hyperpolarize a cell, and then it repolarizes back to the resting membrane potential. All right, because we know cells like to be at the resting membrane potential. And most cells, most neurons, that's what they do. When they hyperpolarize, they go back to rest. When they depolarize, they go back to rest. Right? Cells like to go back to a resting state. What's interesting about these cells is they have a rebound. What that means is that when they are habituated, when they're hyperpolarized, and that hyperpolarization goes away, when they habituate to it, rather than going back to the resting membrane potential, they actually depolarize more. So basically, after inhibition, they depolarize above the resting membrane potential, and that brings them up to threshold. And when they get up to threshold, they fire action potentials. So that's really the key to this rebound. Instead of going back to rest, so instead of going back to the resting membrane potential, the cell depolarizes and fires an action potential, which then inhibits the other cell. And then it's going to habituate and rebound and fire action potentials, which inhibits the first one again, which habituates and rebounds, which inhibits the second one, which habituates and rebounds. And in this fashion, they basically go back and forth, taking turns firing action potentials. All right. Does that help it make more sense? Yeah, a little bit.
So the habituation
Yes.
The habituation is them getting used to the signal, right?
Correct. Yeah.
Okay. And then the rebound is a direct result of the habituation. They
Yes.
get used to the other action potential coming in, and do they use that other action potential, or?
Well, okay. Maybe this will help. You guys do not need to understand this at this level to describe this on the exam if you were to have to talk about this, okay? But let's talk about what's happening. With the habituation, with the inhibition, if you remember, when we try to hyperpolarize a cell and make it more negative, there were two main ion channels that we would open to do that. What were the two types of ions that we could make a cell more negative with?
Chloride channel?
Chloride would be one of them. What would the other one be?
Calcium?
Not
Sodium.
calcium,
Sodium?
not sodium. So what does that leave us with?
Potassium.
Potassium, exactly. And it turns out that's what happens here. On these cells actually are dynamic in the expression of chemically gated potassium channels. So what happens is, on first, let's just put 3 of them here. There are 3 potassium channels. So let's say this is our excitatory or our inhibitory. Sorry. Spiratory center. All right. Our inspiratory fires action potentials. And of course, when it fires action potentials, it releases neurotransmitter. That neurotransmitter is going to bind to these potassium channels. And when it binds to potassium channels, potassium leaves the cell. And as potassium leaves the cell gets more negative. That makes sense? Excellent. All right. So here's what's interesting about this cell. As it starts to lose a lot of potassium, what it will actually do is it will actually withdraw the potassium from the channels from the plasma membrane. So what happens is it endocytoses these. It grabs this and brings it in, and grabs this and brings it in, and grabs this and brings it in. Now notice when I do that, my inspiratory center is still releasing neurotransmitter, but can that neurotransmitter affect this cell anymore?
No.
No. So now the neurotransmitter cannot affect the cell. So the cell has habituated. That neurotransmitter is no longer going to have an effect on it. But notice what this cell did. This cell removed potassium channels. Potassium normally leaves the cell. But if I get rid of those channels, more potassium stays in the cell. And of course, potassium is a positive ion. So if more positive ions stay in the cell, what happens to the cell? The cell gets more positive. And that is the rebound. So basically what happens is these cells are dynamic in their expression of their channels. That is why they're able to habituate. And that's why when they habituate, they become more positive as a result. Now, again, you're not going to have to explain this on the exam. I guarantee that. As long as you understand those concepts of inhibition and rebound, know the two areas that are involved in it, that's really the level that I'm looking for. But hopefully taking the time to explain this helps at least to make it make more sense as to how this could possibly work.
So you're just wanting us to know the difference between like the breathe in, breathe out, don't breathe in, don't breathe out, and then the signals that are like the checks and balances of don't keep breathing in, don't keep breathing out.
Absolutely.
Okay.
All right. Great question. Any more? What's next? Not anymore. You definitely have more. What's next?
I was having a hard time with the respiratory control centers, the DRG and the VRG. I kind of was just getting lost during that part.
So perfect. This goes right along with what we were just talking about. So let's actually, do I have that? Yeah, let's bring that up. Which picture do I want? I want this picture. Okay. So everything that we were just talking about, all of that, those two nuclei that play a role in bouncing the signal back and forth to get the diaphragm to contract and relax. All of that is occurring right here. Let's use the highlighter to do it that way. No, it's already blue. Right here in this ventral respiratory group. So there are three main regions that contain neurons that are involved in controlling respiration. We have some up here inside the pons. And we have some here inside of the medulla. Okay. In the medulla, notice there are two main clusters, the ventral respiratory group and the dorsal respiratory group. The ventral respiratory group, as we mentioned, is the one that sets the primary resting rate, right? What we call the eupnea. But as we've talked about, that is great if you are maintaining homeostasis in your body, but all sorts of other things could be going on. You could be exercising, you could be eating, you know, your body's conditions are changing constantly. So one of the things that happens here in our dorsal respiratory group is our dorsal respiratory group, all of the areas receive a lot of information, but here in particular in our dorsal respiratory group, we receive a lot of information on the internal conditions of the body. What are the oxygen levels? What are the carbon dioxide levels? What are the pH in the blood? All of that type of stuff. And based on that information, we can then modify what is happening in the ventral group. So that chemical information comes in. As it comes in, and it can be coming in from the blood, it could be coming in from the interstitial fluid, from the cerebral spinal fluid, right? All this information is coming in, and we're sending that and modifying our breathing rate based on that. But go ahead.
The dorsal receives and modifies
The dorsal receives and modifies the ventral. So it takes the information and then tells the ventral how to change its behavior based on that. The pons basically makes changes. Well, it does two things. One of the things is, again, this circuit isn't perfect. So one of the functions of the pons is to maintain smooth control of this. People who actually have damage to the pontine regions, pontine respiratory centers, can end up with a very distinct breathing dysfunction where they start taking longer and longer exhalations as a result of that. So it can be disruption. So it helps to maintain the smooth control of that. But it also modifies breathing based on not so much what's happening internally in the body, although that can be part of it, but based on what's going on with the body. If you are exercising, if you are scared, if you are warm, if you are cold, right? If you're hungry, or if you are consciously deciding that you want to speak. Obviously, changing the movement of air so you can produce sound is important. Or you may just like the idea of taking a really big breath. Right? So that voluntary, that volition we talked about. So that basically what's going on with the body, both internally and externally, if you're exercising or if you're angry, those things can influence and modify breathing as well. So these are the three basic areas. And again, you got to remember, or because you're stressed, absolutely. All of these processes are more complicated than we were talking about. So I'm doing a lot of hand waving here because I don't want to take the time and energy to go into the in-depth that is necessary to explain this. So as long as you understand these three basic categories and these three basic functions of these three areas, that's really what I'm looking for. All right. I mean, yeah, we could spend a whole week just talking about, heck, we could spend 3 weeks talking about the control of respiration. This is an incredibly elaborate system, but at its core, these are the main components. So I appreciate your frustration and confusion on it because it is challenging because, yeah, I'm purposely not going in depth on this. What is going on? Oh, the silver alert, missing elderly person. All right. I hadn't heard that before. So yeah, I don't have any idea what I was saying before that. All right, excellent. Next question.
Okay, wait, so just to clarify, just know and understand what those respiratory centers do.
Yes, understand the basic components of what they do and the basic, and again, in that ventral region, the basic functions of the inspiratory and expiratory center. Right. So as long as you have a basic understanding of how these things work, that's all I'm looking for.
And so like a question for that would be, what are the control centers of respiratory? What are the
Sure.
respiratory?
Identify the individual respiratory control centers and describe their basic function. Sure, that would be something. Absolutely. All right. Or, describe reciprocal inhibition and explain how it plays a role in respiration or something along those lines. Sure. I mean, those are things that could be possible questions. These are the things that also, again, some of the things that aren't emphasized as much also end up, multiple choice questions and things along those lines as well. So there could be ways that it could be related to that way as well. I'll be honest, I don't remember if they're what essay questions are on that control. There's got to be something, but I don't remember what they are off the top of my head. I haven't had a chance to, I'm still grading my 430 exam, so I haven't had a chance to look at your exam yet to see what modifications I have to make. But what I would say is, obviously, anytime there's not as much detail, even if there is an essay question, it's going to be one of the, 3 or 4, 5-point essay questions as opposed to the, 8, 9, 10-point essay questions. Anything else?
Regarding histology of like the bronchioles, do we need to be able to identify primary, secondary? How specific do we need to be with that?
So primary are easy to distinguish because they're going to be lined with the ciliated pseudostratified columnar. Bronchioles are going to be easy to identify histologically because there's no cartilage and they're lined with a simple cuboidal epithelial tissue. And then obviously the respiratory ones have that and alveoli on them as well. Anything in between primarily is going to have simple columnar. And so it's really going to be hard to tell the difference between a secondary, tertiary, quaternary, and so on and so forth. So probably just bronchiole would be fine at that point. I mean, bronchi, sorry, would be fine at that point, but I wouldn't probably expect anything more than that. I mean, the top end, you need to be able to tell. That's easy because the ciliated pseudostratified columnar. The bronchiole are easy to tell. Everything else in between is basically just a bronchi, because histologically, I don't expect you to be able to tell the difference between a secondary and a tertiary, right, or something like that. Now, again, on one of those models, like the bronchial tree, can we easily tell which is which? Yeah, absolutely. But histologically, no. All set? Well, it is Wednesday, I will remind you. So obviously I have office hours today after class, office hours tomorrow. I'm always available by email and stuff like that. So if more questions do come up, then please, by all means, take the time and ask them. Thank you guys for sticking around. I always find it ironic that the ones who seem to do the best on the exam are the ones that stick around for the reviews. So hopefully that bodes well for all of you. Excellent. All right. You guys have a great day. Have a safe day. Have a good weekend. And I will see you guys, I guess, next Wednesday. Hopefully, again, I am available if you have any questions. But if everything goes well, then I'll see you again in a week. All right. Bye. Good luck.